And in this lecture, we're covering organic molecules. This is the second video lecture, and we're gonna start with organic molecules. Yeah, sounds sounds right. But the first part covered matter, energy, chemical bonds, and some important inorganic molecules such as water, salts, acid, bases, that also important for normal function of living organism, right? So make sure you watch the first part. And today we're gonna start with organic chemistry. It's gonna be like really brief introduction. So nothing difficult, nothing you need to worry about. Okay, so as soon as I will find that slide where I stopped last time. Okay, I know I covered this already. I covered buffers. So here it is, organic compounds. So let's go ahead and begin. Organic compounds contain carbon, except carbon dioxide, carbon monoxide, which are inorganic. They're unique to living organisms and include carbohydrates, lipids, proteins, and nucleic acids. So again, organic compounds are not limited to these four groups, right? We have many other type of organic compounds, but for biology, this is the four groups we are specifically interested in. They are macromolecules. So many of them are polymers. Well, many of them, all of them, carbohydrates, proteins, and nucleic acids, except of lipids. Lipids are not polymers. Now, what does it mean, polymer? Polymers are chains of similar units, monomers, or what we call them sometimes building blocks. So if you have repeating similar units that connected through covalent bonds, then you can synthesize polymer from monomers, right? Now that reaction that synthesize polymers called dehydration synthesis, and you can break polymers back to their building units or monomers by hydrolysis reaction. Both dehydration synthesis and hydrolysis tells you that water will be involved. Now synthesis, you're gonna lose water, right? So water will be a product of the reaction. That's why we call it dehydration. And hydrolysis, water gonna be reactant. So you're gonna use water to break chemical bonds between monomers. Okay, so on this slide, you can see how this dehydration synthesis and hydrolysis work. So dehydration synthesis tells you you're gonna make something bigger. And here's your monomer one that has this hydroxyl group over here and hydroxyl group over there with a monomer 2. So, so that will combine to create water, and monomers will be linked by covalent bonds. So monomers are joined by removal of OH from one monomer and removal of hydrogen or H from the other at the site of bond formation. Now hydrolysis, here we have two monomers already joined together, and monomers are released by the addition of a water molecule, adding OH or hydroxyl group to one monomer and hydrogen to another. So you add these groups to monomers and you release them. And here we have example of glucose and fructose. Those are monomers of simple sugar, and cells. Well, those are not human cells. This will happen in plant cells, but cells can combine two monomers and remove water to form covalent bond, right? So here, sucrose. So sucrose is your table sugar. At the same time, this is a reversible reaction. Now, sucrose can be broken into glucose and fructose by adding molecule of water. And this happens in your GI tract. So plants make sucrose for you, right? Through dehydration synthesis, and then through hydrolysis in your GI tract, you release those monomers from sucrose, right? So that's what we call digestion. So you digest sucrose into glucose and fructose, and then you absorb those molecules into your bloodstream. Okay, so the first group that we will cover today is carbohydrates. Includes sugars and starches, contains carbon, hydrogen, and oxygen. And it's in a specific ratio. So you have as many oxygen as you have carbon, right? So carbon and oxygen, and then you have 2 times more hydrogen. So we say hydrogen to oxygen is 1:2 ratio. There is 3 classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. And major function include energy. So it's a major source of cellular fuel, for example, glucose, and structural materials. For example, ribose sugar in RNA, or in DNA, you have deoxyribose. So not only you use sugar as a source of energy, and carbohydrates are preferred source of energy for your cells, but you also use sugar to make a hereditary material such as DNA. So monosaccharides, the first group of carbohydrates, that they are simple sugar containing 3 to 7 carbon atoms. So here's their universal formula. So it's carbon and water, right? And then you take it n times. So to be a monosaccharide, this n must equal 3. It can be 4, 5, 6, up to 7. So here's examples of monosaccharides. You have glucose, fructose, galactose. Those are hexoses. So the hexoses have 6 carbon atoms, and pentose sugar with 5 carbon atoms, deoxyribose, sugar of DNA, and ribose, sugar of RNA. Now, glucose, fructose, and galactose, they are isomers. That means that chemical composition is the same. It's C6H12O6, but they have different structural characteristics. So structure is different. That's why they isomers with the same formula, chemical formula. Disaccharides are double sugars, and they are formed through dehydration synthesis. They're too large to pass through cell membranes. That means for you to absorb sugar, you need to break disaccharides into monosaccharides first, right? And that's what happens when you digest disaccharides. So here's some examples. Sucrose, that's your table sugar. You have glucose and fructose joined together through covalent bond. Then maltose is actually glucose, 2 molecules of glucose that connected. And lactose, this is the one that you will find in milk, is galactose and glucose. So you see all 3 have glucose. Right, and the second sugar is different, is a fructose, another glucose, or galactose. The disaccharides are also isomers. Polysaccharides are polymers of simple sugar. For example, starch starch is found in plants, and glycogen found in animals. Now, they call glycogen sometimes animal starch, right? Because we don't have starch stored in our cells and organs, but we store sugar in the form of glycogen. And by the way, this word glycogen means, you produce something, you generate, right? It's a glucose generator, so you can make glucose from glycogen. Polysaccharides are not very soluble. So here's the example. Here is the glycogen. Polysaccharide is a simplified representation of glycogen, a polysaccharide formed from glucose unit. So you have a glucose unit joined together. And polysaccharides are made of long branching chains, polymers of linked monosaccharides. The second group of large molecules, organic molecules, are lipids. Now look, lipids, similar to carbohydrates, consist carbon or contain hydrogen and oxygen, but less oxygen than in carbohydrates. Remember, in carbohydrates, we have as many oxygen atoms as we have carbon. Here we have way less oxygen. Sometimes phosphorus can be part of lipids as well. All lipids are insoluble in water, and we have many types, like neutral fats or triglycerides, phospholipids, steroids, isocyanoids. So here's the first group, triglycerides, neutral fats, solid fats or liquid oils composed of 3 fatty acids bounded to a glycerol molecule. So here's our glycerol molecule. Here's the 3 fatty acids. And when we join these 4 molecules together, we make triglyceride or neutral fat. And of course, water is released, so dehydration synthesis. And now you can, you see the 3 dots over here means there is many of these CH2 groups in between. So you can see we have so many carbon and hydrogen and really just few oxygens. Now, the main function is energy storage, insulation, and protection. Now, triglycerides are just macromolecules. They're not polymers because they're not made of repeating units. Now, when we look at this fatty acid, so let's go back again. So here's this fatty acid. They all have this carboxyl group, COOH. Now, those fatty acids can be saturated fatty acid and unsaturated. Now, saturated, they have single bonds between carbon atoms and maximum number of hydrogen. So they saturated with hydrogen. They include solid animal fats, for example, butter. So actually any fat, not just animal fat, but fat that is solid at room temperature contains saturated fatty acids. Unsaturated fatty acid, 1 or more double bonds between carbon atom. So if you look over here, you see this double bond between carbon, and because we have a double bond, we have less hydrogen. So we are not saturated of hydrogen. So we reduce number of hydrogen atoms. An example can be plant oils, for example, olive oil. Now you also can look over here that here we have double bond. And so it's unsaturated, so that's obviously unsaturated fatty acid, and it has this kink, so it's not straight linear molecule. Now this molecule below also has a double carbon bond, so it's also unsaturated, but now look, it's a straight molecule. Now why would it be important? Because those molecules that are unsaturated, but they still have this linear structure, and hydrogen are across the double bond, those are trans fat. And trans fat happen to be the worst type of fat for human cells and human health. Now, this is cis double bond when we have hydrogen on one side of double bond, and those are healthy unsaturated fatty acids. Okay, so unsaturated fat, the so-called good fat, can be found in nuts, avocados, other vegetables. The molecular structure of unsaturated fat causes it to be lower in calories than other fats. So molecular structure of unsaturated fat shows spaces available for hydrogen atom to bond, right? So we have less hydrogen atoms over here. Saturated fat, those fats are found mostly in animal products. It is recommended that people reduce their consumption of saturated fat in order to stay healthy. In a molecule of saturated fat, every space for hydrogen is filled. That's why it's saturated with hydrogen that we already mentioned. And here's our trans fats. So trans fats are unsaturated. So it's unsaturated, or what we call good, but we put it in a parenthesis because nothing is good and bad, really. It's just too much or too little of the nutrients can be bad or good for you, right? So trans fat are unsaturated fats which have been partially saturated with hydrogen to extend their life shelf. Unfortunately, these trans fats are found to elevate cholesterol and should be avoided. And I specifically didn't use this word bad cholesterol. It's just low density. It's low density. Okay, I forgot what it's called. Yeah, that's what we call. Let me just stop for a moment and find it out for you. Okay, so I find out it's called low density lipoprotein. That's what we call a bad cholesterol. All right, so that's what happened when we buy a French fries or processed food. The processed food, when they process food, it's tend to increase the amount of trans fats. Another type of lipids are phospholipids. Those are modified triglycerides. So remember triglycerides when we have glycerol, right? And 3 fatty acids attached. In phospholipid, glycerol are attached to 2 fatty acids and a phosphorus containing group. So that's called phosphate head, and those are called tails. So head is our phosphate, and tails, regions, have different properties. Phospholipids are important in cell membrane structure. Actually, if you look at cell membrane, it's made of double layer of phospholipids. Now, what are those different properties? Well, the phosphate group is a polar part. It's a polar head. And if you have a polar molecule, then this molecule will be attracted to water. So it's a hydrophilic part. And the tails are non-polar molecules. If we have non-polar molecules, it's hydrophobic. It will repel water. So now, because you have, like, for example, that's your, that's a simple representation of a cell. You have fluid outside the cell and you have fluid inside the cell. Because of the properties of phospholipids, the heads, phosphorus-containing heads will oriented towards the water because they love water. They're attracted to water. So that's why we're forming double layers. So one layer, see how we have heads facing water outside, and in a layer, we have heads facing a liquid of water inside a cell. And that's important property of these phospholipids that pretty much allow us to form this biological bilayers, the membrane. And without this property, we would probably never had cells on our planet. And if we wouldn't have cells, we wouldn't have living organism, because cell is the structural unit of any living organism. So life exists on this planet because of phospholipids. Steroids are another example of lipids. They interlocking 4-ring structure. So here you can see 4 rings. So 1, 2, 3, 4. And then we have some different tails over here. Example of steroids include cholesterol, vitamin D, steroid hormones, such as testosterone, estrogen, progesterone, and bile salt. Now, cholesterol is the basis for all steroids formed in the body. And eicosanoids, I think I pronounced it wrong the first time. So is it easy to pronounce them eicosanoids or eicosanoids? This is not really the word that I use, because kind of like the example in biology, example of this chemical are prostaglandins. And I like this name. Prostaglandins, those molecules was named after prostate gland. You know, like a prostate gland that is inferior to urinary bladder and it surrounds male urethra. There is a gland called prostate gland. And those molecules were discovered in that organ. That's why they named them prostaglandins. So they derived from a fatty acid in cell membrane. Now, why these prostaglandins are important? Because when cell is injured, right? So here's a human cell, for example, and we have injury. Now, this injury can be a cut, or it can be bacteria that damage a cell. Then the membrane is made of phospholipids. Remember, so phospholipids in the cell membrane are converted into prostaglandins. As it says right there, the previous slide that says that derived from a fatty acids in cell membranes. And phospholipids, remember, they're made of glycerol and fatty acids. So when we have this conversion, prostaglandins are formed, and they are actually inflammatory chemicals. So they will cause inflammation, and the sign of inflammation include redness, worms, swelling, and pain. Now, if we can block this conversion, right? So for example, we still have bacterial infection and bacteria damaging our cells, and we have very severe inflammation. And one reason, it's not the only reason, but one reason why we have inflammation, because we have prostaglandins now. What if we can block this conversion? And then we reduce the amount of prostaglandins so we can reduce inflammation. And we can do it using this medication, aspirin and Advil, block this conversion and called anti-inflammatory drugs, right? So interesting how, you know, scientists learn the mechanism of action of this medication. So it's not just, okay, take Advil because somehow it reduces your inflammation. Well, it's wonderful that we can understand what's really happening, how exactly this medication help us to reduce redness, worms, swelling, and pain, right? That's a use of antibiotics and blocking of the enzymes, because you need enzyme to convert phospholipids into prostaglandins. So when you block that enzyme that catalyzes conversion, then you reduce the amount of prostaglandins and signs of inflammation. Other lipids include vitamins A, E, D, and K. So it's called ADEC. Right, so that's lipid-soluble vitamins, lipoproteins that transport fat in the blood. So actually here, our high-density lipoproteins and low-density lipoproteins. And what's this? lipoproteins do, they deliver cholesterol. Now look, so for example, that's your cell, right? And you have fat accumulating in the cell. Now, let's say it's a cholesterol, right? So you have accumulation of cholesterol. So you have lots of cholesterol in the cell. Now, what are you gonna do? You have, it's a special protein that's represented here as a truck. This protein removes excess cholesterol from the cells and deliver it to liver. And liver converts this HDL into bile salts. Now, what this transporter does, low density lipoproteins, they transport the cholesterol back to the cells, right? So so that's why we call this a good cholesterol, and we call it a bad cholesterol. But cholesterol, the molecule of cholesterol is not changing. It's always the same cholesterol. That's what, you know, it's important to understand, and that sometimes we forgot. We just somehow think that bad cholesterol might have a different structure from good cholesterol, but no, it's not. Cholesterol is always the same. This transport lipoproteins that either transform cholesterol to the liver, and that's good, or the other transform cholesterol to the cells, and it's not good for our health. Okay, so here's, we continue talking about lipids, steroids, and this example, that is vitamin D, calciferol. It's made from cholesterol in skin and requires sunlight for that, secreted into bloodstream, and vitamin D increases absorption of calcium in small intestine. What is this number 2? Oh, that's supposed to be next to calcium. And vitamin D is added to milk in US to promote calcium absorption. And now we are in the next group of organic macromolecules, proteins. Proteins are polymers of amino acids, 20 different types of amino acids. They join by peptide bonds. Proteins contain carbon, hydrogen, oxygen, nitrogen, sometimes sulfur, and phosphorus. So if you look at different amino acids, remember we have 20 to make proteins. So all amino acids, their generalized structure include a central carbon, then we have amino group attached to it, carboxyl group or acid group, and hydrogen. So this part shown in the oval is the same for every amino acid. And remember, carbon can form 4 bonds. So the last over here, well, it doesn't necessarily last, one of the bonds attached to what we call functional group or R group, and this R group is different. And the properties and names and chemical characteristic of the amino acid depend on this side group or R group. So if you just have hydrogen over here, it's glycine. It's the simplest amino acid. Then we can add, you know, other structures. So we're adding other structures here, and we get a different amino acid like aspartic acid, lysine, cytine, and so on. So that's how different amino acids are built. Then inside your cells, those amino acids binds to each other. This is called protein synthesis. It's a very complicated process. It includes DNA, RNA, messenger RNA, transport RNA, ribosomes, genetic code. So it's all this fun stuff that we are not covering over here. But if we look at it from a simple point of view, it's just this dehydration synthesis reaction. When one amino acid is attached to another amino acid, a molecule of water is removed, and from a monomers, we're forming polymers. So this is dipeptide over here because we have two amino acids joined by peptide bonds. So the acid group of one amino acid, so that's acid group, is bonded to the amine group of the next with loss of water molecule. And of course, it's a reversible reaction, and this is your digestion. So you also have enzymes that break down this peptide bond, and it's a hydrolysis. So peptide bond linking amino acid together are broken when water is added to the bond. Now, proteins are very complex molecules. Not only your cells need to bind amino acid together, that's just the beginning of the process of protein synthesis. So when just amino acid attach, we call it primary structure of proteins. It's a sequence of amino acid that form the polypeptide chain. Then, because this amino acids have all these different functional groups or R groups, we have lots of interactions between these R groups. It's a hydrogen bonds formation. And then our primary structure folds on it all. So it's, here we get the secondary structure. So the primary chain forms spirals. We call it alpha helices, and sheets. Call it beta sheets. So alpha helix, the primary chain, is coiled to form a spiral structure, which is stabilized by hydrogen bonds. Beta sheet, the primary chain, zigzags back and forth, forming a pleated sheets. Adjacent strands are held together by hydrogen bonds as well. But that's not enough to make a functional protein yet. Because the protein must gain its tertiary structure. So it's superimposed on secondary structure. Alpha helices and or beta sheets are folded up to form a compact globular molecules that are held together by intramolecular bonds. So the tertiary structure of a prealbumin, a protein that transports the thyroid hormone thyroxine in serum and cerebrospinal fluid. So here's the example of tertiary structure. When you see we have the alpha helices and beta pleated sheets that arrange in a precise three-dimensional shape. Now, some proteins, not all proteins, but some also have quaternary structure. When we have two or more polypeptide chains, right, in the first one, we have one polypeptide chain. So it's just one polypeptide chain that is in a three-dimensional configuration. Here we have two or more polypeptide chains, each with its own tertiary structure. Combined to form functional protein. So quaternary structure of functional prealbumin molecule. Two identical prealbumin subunits joined together to form the dimer. So here we have like two polypeptides, right? Each one has the tertiary structure, but when two combine, that's our quaternary structure. Another example, I don't remember if I have it or not, but yeah, I have it over here. That's a hemoglobin. And hemoglobin, actually, we have not two, but four polypeptides that join together. So this is a quaternary structure of hemoglobin. Now, proteins can be divided into globular or functional protein and fibrous or structural proteins. So globular are compact, spherical, water-soluble, and sensitive to environmental changes. Specific functional regions are active sites of a protein. An example, antibodies, hormones, enzymes, chaperones. Well, the chaperones, yeah, so they are proteins. Chaperones are proteins that kind of help a protein. So they guide proteins along the proper pathway for folding. That's what molecular chaperones are. But those are all globular proteins. Now, fibrous proteins, they are strand-like. They are water insoluble and stable. Now, example would be keratin, that is a major protein of your hair and your nails, elastin, collagen. Those are proteins that found in your dermis of your skin, and certain contractile proteins, such as like actin and myosin in your muscle cells that allow your muscles to contract. Right, so you can see on this diagram, even it's, of course, it's just a diagram. It's not real structure of a protein, but here a collagen that is strand-like, water-insoluble protein found in your skin, for example. And here's the hemoglobin, the protein that found inside your red blood cells and deliver oxygen to your cells. Oops, I'm sorry. Now protein, just to make sure I didn't skip anything, protein denaturation. It's a shape change and disruption of active sites due to environmental changes. For example, change in pH when we increase acidity or increase in temperature. So when we change pH and temperature, proteins can lose their shape. And when they lose their shape, they cannot function anymore, right? Well, denature can be reversible and irreversible. Reversible is in most cases if normal conditions are restored. Irreversible if extreme changes damage the structure beyond repair. For example, if you cooking an egg, right? So egg, this white part is made of albumin, so that's a protein. So you can see how it is different from raw egg, and there is no way you can reverse it back. When you warm the milk, when you just warm up the milk, then I see how that's a protein in the milk and it's losing its change. But then if the temperature is restored, it can return back to the same condition. However, if you boil milk, it's never gonna taste really the same. So you will know a boiled milk. If you boil milk, then it will be extreme changes that will the proteins will be damaged beyond repair. Now, enzymes. Why all of a sudden we talk about enzymes? Because they're also proteins. Enzymes are biological catalysts. They lower the activation energy and increase the speed of reaction. Millions of reactions per minute with enzymes. So here we can look at the reactions that would happen without enzymes. Actually, all reactions that happen with enzymes can happen without enzymes, but it might take million times longer, right? And of course, that speed of reaction is not even close to sustain life. So without enzymes, we're definitely gonna die. There is no way living organism, and I'm not only talking about humans as a complex living organism, even bacteria, even like a small microscopic living organism, they will not survive without enzymes. But what enzymes do, they lose this activation energy and reaction happens quickly. Characteristic of enzymes. They often named for the reactions they catalyze, usually end in A's, like hydrolases, oxidases, hydrolase, oxidase. Some functional enzyme consist of protein, apoenzyme, and cofactor that can be some ions or vitamin. If it's a vitamin, we call it coenzymes. Now, here how enzymes work. So that's a simple diagram, shows us, okay, so here's enzyme. Enzyme has active site, and here we have substrates. For example, it's 2 amino acids. So amino acids are attached to active site of the enzyme. So substrates binds at active site. Enzyme changes shape to hold substances in proper position. Energy is absorbed, bond is formed. So internal rearrangement leading to catalysis occur. So here water is removed and we have a product, right? Product is released, enzyme returns to its original shape and is available to catalyze another reaction. So you see, we started here with enzyme, we ended up with the enzyme. It's not damaged, it's not consumed. What it did, it just lowered this activation energy by changing the shape, but rearranging those substrates and increase the speed of formation of those chemical bonds. Now, why we wanna, obviously enzymes are extremely important, right? But we wanna talk right now about enzyme inhibitors. And then I'll tell you why all of a sudden we wanna talk about it. Enzyme inhibitors are chemicals that prevent a substrate from attaching to enzyme. So here's our normal binding enzyme. So here's enzyme, here's active site, and here's our substrate. So substrate feeds inside the active site. And it's like a lock and a key. When it feeds, enzyme catalyzes the reaction. Now, what if we have chemical right here, shown here in yellow, that is enzyme inhibitor? What if this enzyme inhibitor binds to active site, and then what happens? Then substrate cannot bind. That means reaction will not happen. In this situation, it's a competitive inhibitor because it binds to the same site, active site, where substrate binds. We also might have non-competitive inhibitor. It binds to any other side of enzyme. Another side is called allosteric side. So when it binds to allosteric side, you would say like, okay, well, it's not on the way somewhere here. So maybe a substrate can bind, but this reaction causes changes in a shape of active site, and substrate cannot bind either, right? Now, if we have these chemicals and we can use them, what if we can use these chemicals in the medicine to prevent some reaction in our body to happen? Can we or not? Well, before talking about that, let's say what chemicals do it for us and how dangerous they are. Cyanide, for example, CN. It interferes with enzyme cytochrome excitase in cellular respiration. So we have this enzyme in cellular respiration, and remember, cellular respiration produces ATP. So if we deactivate this enzyme, if we make this enzyme non-functional, that's what cyanide does, then our cells cannot make ATP and cells die. That's why if somebody is poisoned with cyanide, they're gonna die very quickly. Another acetylcholine esterase inhibitor interferes with enzyme in the nervous system and causes muscle paralysis. So it's another inhibitor of this enzyme, acetylcholine esterase, and it causes paralysis in the nervous system. What? And now back to my question. Can we use these chemicals to prevent reactions that we don't want to happen in our body? And actually we can. So here's the enzymes inhibitor used in medicine. Very famous antibiotics. They inhibit bacterial enzymes. So if they inhibit bacterial enzymes, then bacteria cannot function properly. Bacteria will die. That's what antibiotics do. Aspirin and Advil, we already talked about it. They inhibit the enzymes that convert phospholipids into prostaglandins. And remember, when we convert phospholipids into prostaglandins, prostaglandins cause inflammation. So we reduce inflammation. Another very important, Lipitor. If you know somebody with high blood pressure and high cholesterol, they're probably taking Lipitor. Lipitor inhibit as T is missing, the enzymes that converts fatty acid into cholesterol. So now if you're taking Lipitor because your cholesterol is high, well, now you inhibit conversion of fatty acid into cholesterol, so you low cholesterol level in your body. Okay, and another example of proteins include protein hormones. All of the protein hormones must be injected. Otherwise, if you just take it orally, protein hormones, you just digest it. Steroid hormones are not broken apart by digestive system, but protein hormones do. Example of protein hormones include insulin, a growth hormone. Another proteins in your body are antibodies. Well, you have this red blood cells called B lymphocytes, and they can differentiate into plasma cells, and plasma cells secrete antibodies. And antibodies are our functional proteins that inactivate foreign agents. So here we have bacteria, for example, shown, and antibodies are attached to bacteria, to antigen on a bacteria, and inactivate that bacteria. Okay, and the last large group of organic macromolecules, nucleic acids, includes DNA and RNA. Largest molecules in the body, contains carbon, oxygen, hydrogen, nitrogen, and phosphorus. Building block of nucleic acids are nucleotides that compose of nitrogen-containing base, pentose sugar, and phosphate group. So here's our nucleotide, so shown right here. So that's a nucleotide made of sugar. It's a pentose sugar, that means 5 carbon atoms, phosphate group, and nitrogenous base. Now, if we're talking about DNA, the sugar is deoxyribose. If it's RNA, sugar is ribose. But those nucleotides, they are the same, right? So part of the nucleotides is the same. Sugar and phosphate group, always the same in DNA, and sugar and phosphate group is always the same in RNA. But what is different is this nitrogenous base. So we have actually 5 different types of nitrogenous bases, but we have 4 in DNA and 4 in RNA. And they are cytosine, thymine, adenine, and guanine. So these 4, C, T, A, and G in DNA, and we don't have T in RNA. We have cytosine. Instead of thymine, we have uracil, guanine, and adenine in RNA. And if you look at this nitrogenous bases, some have like 1 ring, and we call them pyrimidines, and some have 2 rings, and we call them purines. So purines and pyrimidines. So here's our DNA, or deoxyribonucleic acid, has 4 bases: adenine A, guanine G, cytosine C, and thymine T. It's a double-stranded helical molecule in a cell nucleus, provides instructions for protein synthesis, and replicates before cell division, ensuring genetic continuity. So here's our DNA, and we have our sugar phosphate, right? That forming sugar phosphate backbone and bases. And you see we have bases A, G, T, and A. Now this is one strand of DNA. This is the second strand of DNA. And in DNA, because it's double-stranded helix, the nitrogenous bases, they form hydrogen bonds between them. So here's these dotted lines are hydrogen bonds, and there is a rule of pairing. A always pairs with T, and C always pairs with G. So mnemonic is apples in the tree and cars in the garage. So A with T, C with G all the time. That's our base pairing rule. Now RNA, now we have a different sugar. Instead of deoxyribose, we have ribose sugar, phosphate group, and we have also 4 bases, but we don't have T. Instead of T, we have U. And it's a single strand, one strand. So here's some differences and similarities and differences between DNA and RNA. So let's start with similarities. Say the both DNA and RNA has sugar and phosphate that make sugar phosphate backbone. They both use A, C, and G, adenine, cytosine, and guanine, right? And the rest are different. DNA is double-stranded, single-stranded is RNA. Sugar in RNA is ribose, DNA deoxyribose. DNA uses T, thymine, RNA uracil. Then another important structure, DNA is in the chromosome. It's a structure into chromosome. It stores genetic information when RNA is actually, so RNA, you find RNA in a nucleus and in a cytoplasm. They assemble in the nucleolus of the nucleus, and then you will find them in the cytoplasm. It stores not genetic information, but RNA. We have different types. First of all, we have different type of RNA, like messenger RNA, ribosomal RNA, transport RNA. And RNA is important in the protein synthesis. RNA usually does not last for a long time, and it cannot self-replicate when DNA can self-replicate, and this ensures continuity of life because DNA is replicate itself, so you can pass DNA from one cell to another cell. RNA does not replicate itself. Okay, so let's see if I have something else. Yeah, so I have 2 more slides over here to cover. And we will talk about ATP. Now, ATP is an energy currency and it's nucleotide. So remember, DNA and RNAs are made from nucleotides. And what is nucleotide? It's a sugar, right? It's a phosphate group and nitrogenous group. For ATP, we have sugar ribose, the same sugar we have in RNA. So ribose. We have a nitrogenous base. Specifically, we have A, adenine, and we have phosphate group. So if we have actually just, so ribose and adenine called adenosine. And if we have one phosphate group, like a, like a regular nucleotide, that's AMP, adenosine monophosphate, just one phosphate group. If we have 2 phosphates, it's AMP, ADP, adenosine diphosphate, 2 phosphate. And if we have 3 phosphate group, we have ATP, that is adenosine triphosphate. Now this bond shown between phosphate groups, and they're shown in red because they high energy bond. So this, the last bond has the most amount of energy, right? And so when, remember, we talk about metabolism, and metabolism is divided into catabolism and anabolism. So catabolism is this exergonic reaction. So for example, when you eat food, right, you digest food, you break it down, it's exergonic reaction. So that energy that is released, part of this energy will be used to synthesize ATP. And then you use this ATP to carry on anabolic reaction, for example, to make your muscles bigger. So you use this energy, and now instead of ATP, you have ADP. So you break this bond, you use this energy stored in the bond between phosphate group to, I don't know, to grow or to contract your muscles. Now you need to reattach this phosphate group back, and this comes from catabolic reaction. So think about ATP as a rechargeable battery. So that's a battery that's full of charge. You use it, and now your battery is out of charge. Now you need to charge it again. And by saying you need to charge it, you need to attach this phosphate group back to ADP, make ATP, and here you have your functional battery. Again, you can reuse it, right? So here's what the function of ATP. Phosphorylation. When the terminal phosphates are enzymatically transferred to and energize other molecules. Such primed molecules perform cellular work or life processes using the phosphate bond energy. So here, transport work, for example, when we use ATP, ATP phosphorylates transport proteins. So it's attached this kind of like phosphate group. And because of that, this transport protein changing the shape and allow molecules to move and move in and out of the cell. Then mechanical work. ATP phosphorylates contractile proteins in muscle cells and allow muscle cells to contract. Or chemical work, when ATP phosphorylizes key reactant, providing energy to drive energy absorbing chemical reaction. So what this ATP does, so when you break down this bond between phosphate groups, right, you transport this phosphate group to another molecules, and you energize those molecules, and they can do different functions, right? Or cells can contract, for example, or proteins can change their shape, and it's allow solutes to move in and out of the cell, or even, you know, some bigger maybe material. Or you energize different reactants, and the chemical reaction happen, right? So that's how your cells use energy of ATP, that is nucleotide, that is related to nucleotide of DNA and RNA, and use this ATP as an energy currency. Okay, so that's it. We finished this chapter. This is chapter chemical level of organization in biology. So thank you for watching, and I hope it was helpful.